Welcome everybody, and thank you very much, Erika, for being with us for this webinar. It's really a pleasure to meet you again and to have you with us. I will just make a brief introduction and then Erika will give the lecture. But just to remind you, because some of the attendants are not members of the Global Virus Network, and we very much welcome them. So just to remind everybody that the Global Virus Network is a really science-driven network created by Bob Gallo and Billy Hall and Dr. Reinhardt a few years ago, really aiming to merge the best experts in virology worldwide. We have now about 55 centers and 10 affiliate centers all over the world in 33 countries. So this is really a science-driven network, very informal, very nimble, where we have joint scientific activities, in particular organized through an annual meeting, which gather all the GVN directors. We have task force on specific viruses. We had task force on Zika virus, chikungunya, but also HTLV1, and now obviously COVID-19, which is at the forefront of our activities. The GVN covers all human viruses, and this is really a very important point. We have also a lot of educational training activities, training the new generation of virologists, and this is really a very important and sometimes underappreciated activities. We have also advocacy activities, which have been increasingly important. So in this context, and I very much thank Lynn Manley, the new Vice President of the GVN for this. We have initiated this series of webinars, and it's a pleasure to have again Erika Ollmann Saphire. Erika is a highly recognized scientist, and she has a very interesting combination, a blend of conducting basic research, very much focused on structural biology. She is one of the investigators having in hand the Titan Cryos microscope, and with this really deciphering the interaction between viruses and the host immune response, with a lot of very important contribution regarding Ebola, Lassa, so hemorrhagic fevers. And so Erika has been leading a very interesting consortium supported by the NIH, combining different centers worldwide, including in Africa, to study these hemorrhagic fevers. She is now a professor at La Jolla Institute for Immunology. And regarding COVID-19, actually she is in charge of a very important consortium supported by the Bill and Melinda Gates Foundation to really investigate the value of different antibodies possibly being used for therapeutic and for treatment of COVID-19. Erika has always provided a great support to the Global Virus Network, and her activity has been recognized in 2018 by the GVN Robert Gallo Award for Scientific Achievement. So it's really a pleasure, Erika, to have you with us, and we look forward to your lecture, around 35-40 minutes, followed up by questions. Thank you very much.
Thank you very much, and it's a pleasure to be with you today. So today I'm going to tell you about our global consortia for antibody therapeutics. It's called COVIC, the Coronavirus Immunotherapeutics Consortium, and it's now funded by two equal partners, the Therapeutics Accelerator of the Bill and Melinda Gates Foundation, MasterCard Welcome, and private philanthropic support, and a supplement from the National Institutes of Health to bring in complementary analysis that we felt that we needed. Now our goals are two. The primary goal and the mission of the Therapeutics Accelerator, as set forth to me when they called and asked if I would do this, is to find one or more antibody cocktails, fully human neutralizing antibodies, probably 2 in a cocktail, with activity against SARS-CoV-2. And the interest of the foundation here is to make sure that we can save lives in low and middle income countries, that people in these places are not priced out of an antibody therapy. because we have seen very clearly this year, as in other years and other outbreaks, that virus anywhere can rapidly become virus everywhere. Unless we are able to achieve immune protection everywhere in the world, the virus will continue to boomerang around. But in order to find antibody cocktails that we can mobilize to save lives in low and middle income countries, we have some special considerations. They have to be highly potent. If they're highly potent, we don't need as many milligrams, and what you can manufacture can treat more people. They have to be resistant to escape. We need to achieve a lower cost of goods, and that can be a challenge for an antibody therapy. Potency helps. If you don't need as many milligrams to achieve the same effect. And also durability, manufacturability, scalability. Now, I'm an academic professor, and so while the goal set out forth for me is very much a translational research goal, look at the therapeutic antibodies being raised by all of the discovery engines around the world and find among them one or more cocktails that we can mobilize to save lives, I think it's also an opportunity to gain some information. If we have the 70 different antibody discovery engines now unified into a single effort, with all of these excellent therapeutic candidates in the same place, we have an opportunity to learn something. We could make a very broad, deep, and informative survey of the landscape of antibody activities against the surface spike protein of the virus. Which antibodies are most effective and why, and what makes them effective? Which assays against this new virus best forecast in vivo protection? What else can we learn, and what arsenal we can build so that if the virus changes seasonally, or if, God forbid, SARS-CoV-3 emerges, we have something at the ready. And so the goals are those: a translational goal and a basic research goal. Now, the highest priority use case here is to save lives. And that would be treatment. That would be treating individuals with mild to moderate disease to prevent them from progressing to severe disease. And so this is where antibodies really shine. So just like anti-venom, where earlier is better, the same thing with antiviral antibody. If you find cases that may have underlying immune complications, or they are elderly, or they fit the profile of someone that might progress to severe disease, early antibody intervention might knock down that viral load and prevent a negative outcome. We can also use these antibodies for prophylaxis. Until there are vaccines, you can use them to protect, for example, frontline medical workers or vulnerable contacts, high-risk groups like pregnant women, from becoming infected. And you can use antibody where a vaccine hasn't become available yet, or in an emergent situation where it might not have time for vaccine immunity to develop, to circle around an outbreak situation. So for example, if virus erupted in a nursing home, you could treat the other residents and the staff to prevent spread in that location. Now, I was asked to run this large consortium for coronavirus because of our experience doing a very similar thing for Ebola virus. So let me show you what we built for Ebola virus and why, and what the benefits are of a global collaboration like this and like the GVN. So this work started as Ebola emerged and threatened to become a larger pandemic. At the time, and this was 2013, the state of the art in antibody therapeutics for Ebola was a human antibody in orange that neutralized beautifully in test tubes but did not protect non-human primates. They all died. And a cocktail, shown in blue and purple, that binds the upper glycosylated regions, including a not yet well visible mucin-like domain of these purple and blue antibodies. And the cocktail did protect non-human primates, but in the absence of any good neutralization in vitro. And so we had a neutralizing antibody that didn't protect, and protective antibodies that didn't neutralize. And we wondered what we were missing. Was it that we just needed a cocktail to mitigate escape by hitting different footprints? Was it something about the FC? Because we saw that when this purple and blue cocktail called MB003 was manufactured in plant cells that make an afucosylated glycan that enhances binding to FCR, that improved protection and lowered the dose. Or did this tell us that the neutralization assays that the field were running were not as predictive as they could be? And so the state of the art of the field at the time was that we were clearly missing information. We didn't fully understand what in vitro predictors would tell us how to best forecast in vivo success. We'd also been working with a very small set of samples. The field had at the time maybe a dozen monoclonal antibodies. That's just not enough to see the rules. Maybe we'd only seen the outliers. We needed more antibodies and better antibodies. The other thing is we needed more expertise. Everybody was working in a silo with 1 or 2 labs working individually, and they each had their own flavor of an assay. And if you run different assays in different conditions, you will get different results. We also had people of different expertise that weren't working together. And if there was information we were missing, we needed to draw upon those other brains in order to answer the question. And we had a pandemic that threatened to escalate. And so we, as a international research field, needed to be smarter about how we went about this. We certainly needed to work faster. We needed to find a way of working together. Now, it makes perfect sense that we should do all of these things. We should always do these things. We should be smarter. We should work faster. We should work together. But the challenge was in trying to figure out how to do all 3 of those things at the same time. Because the time required to organize into a group can cost you if a pandemic is growing. If you've organized into a large group, you can stifle individual opinion. You can lead to groupthink. If we are looking for individual voices and new ideas, how do we find those and celebrate them? How and learn whether or not they're useful? How do we gain new information that we hadn't had before? The other issue is that it's the fundamental idea about the large collaboration. You know, we've all grown up as competition as the model in scientific research, because competition is what drives us to get results faster. Competition is what drives innovation. We need that innovation in order to find new solutions, new information, better samples. And we wrote a grant to fund a large collaboration to find better antibody therapeutics for Ebola and to do better assays for Ebola and see what we could figure out. And reviewer 2 had some valid criticisms, as it really is not possible to organize such a large effort. And at that point in time, we were proposing a group of 12 different labs. We ultimately grew to 44. Reviewer 2 thought that the organizing effort would be too slow, that we would not learn anything new than any individual lab racing forward by themselves would, and that it fundamentally violated the need for competition in order to work faster and find new and better things. And so we took that as constructive criticism, and we built a research model that we felt could do all 3 of those things at the same time, the smarter, faster, and the together. That we worked to find frameworks and legal documents that would protect intellectual property, that would allow the benefits of collaboration and the benefits of competition to coexist at the same time to get the best of both. In order to get that broader set of samples and that broader expertise and the side-by-side comparison of all of the different antibodies now being raised against Ebola virus, and to do it at a larger scale with more people and yet faster than we had done before, we needed to figure out a better research framework. One part of that framework, which we're using in COVID now, is that to encourage participation, we had to blind the samples. So that in our study, everything was given a code name. And so we would talk as a group about what antibody 45 did and antibody 87. That meant the investigator's intellectual property and unpublished information were protected. He or she was free to publish their own papers on their own antibodies in their own lab, and that we would together would be working on the code names, understanding what antibodies in general would do. That protected their intellectual property and allowed them to participate, because even if the scientists had wanted to join, you know, maybe their university or their company wouldn't have allowed sharing of the unpublished samples. And to address this speed, we set up 2 parallel experimental tracks, a fast track and a comprehensive track, kind of a tortoise and the hare. The comprehensive track was, we're going to gather all of the therapeutic antibodies now being raised around the world. We're going to blind them all. We're going to put them in the same place. We're going to run this very large array of antibodies through everybody's individual assays. Everybody in the field had some different expertise. Some did glycan sequencing, some did protein structure, some looked at FC activities. We had 4 different ways of measuring neutralization. All of those experiments would be done on the same set of the world's antibodies. And then we would look at the end at which antibodies were best in these assays and which assays best forecast protection. So that was a comprehensive track. That was going to take some time. The fast track was a proof of concept. Can we take a couple of antibodies that we think look good? Can we mix and match and get something in place as a proof of concept? And so with that research framework was born this group that went up to 43, now 45 labs across 5 continents, bridging academic, industry, and government fields to do a single comprehensive study to figure out what antibodies against the Ebola viruses are most effective and why. What assays should we do to find them faster? We were ultimately given 230 antibodies from different investigators' labs, and we did all of these different kinds of experiments to figure out everything we could across them. So we mapped epitopes onto the viral glycoprotein. We could see where each FAB fragment attacked the white surface viral glycoprotein. We could cluster them into the ones that had similar footprint. But of the ones that had similar footprint, so for example, you can look at the glycan cap at the top, and there are all those different blue FAB fragments. They hit the same place, but they come at it from different angles and had different levels of success. So we can then tease apart the secondary variables about why within that group, some are better than others. We started with that question of whether neutralization in cell culture, the ability of that antibody to block viral infection in a cell line, could forecast protection in an animal model this way. We measured neutralization in each of 4 different assays, and then we coupled that to in vivo protection beginning in the mouse model. We separated the antibodies by epitopes. So we have a large group against the glycan cap, we have a group against the head, we have a group against the mucin-like domain. And we can separate those into the ones that neutralize potently, in the middle, and weakly in each of these different assays. And you can see we've got different readouts in different assays, as we have all seen in scientific meetings. So across this broad collaboration, what was the relationship between neutralization and protection? Well, we found antibodies that neutralized potently and reliably and achieved protection. That's what we were looking for. That's what we expected. We also found antibodies that could neutralize pretty well, but failed to protect in the animal model for one reason or another. We had antibodies that achieved protection in the absence of any measurable neutralization at all in any assay. So whatever it was those antibodies were bringing, it wasn't being measured in the ability of the antibody to block infection in those cell lines. And then we had antibodies that protected, but mostly didn't neutralize. The only assay in which they measured any neutralization at all was authentic Ebola virus. They wouldn't neutralize any pseudovirus system. And if you had selected those antibodies to go forward based upon their ability to neutralize at all, you never would have picked them. And they were even kind of crummy at neutralizing authentic Ebola virus, and yet they protected. And so we were expecting the ones that neutralized and protected, and we're expecting the ones that failed to neutralize and failed to protect. But the outliers were interesting, and that told us what we've been missing all along. If you look at the outliers, and look at those that protect in the absence of neutralization in red, and those that neutralize but fail to protect in blue, the answer had to do with what the Fc domain of the antibody was doing. If they had natural killer cell activity, they would tend to be protective. If they were missing it, they weren't. If they had more phagocytic activity, they would tend to be protective. If they were missing it, they weren't. So the activity of the Fc was what determined efficacy for those groups. At the end of a couple years, we had a data set that looked like this, where these were all the antibodies we were given, and this is everything we measured about them. And so we could build network correlation models of every variable we measured about the antibody and its correlation with every other variable. And these are on our website. You can play with this at your desk to see, well, which sorts of features tracked with other features. If it had a certain glycan, did it have a certain function? And then we could build a logistic regression model to say, okay, well, of all the things we've done, which was most useful? Which of the things we measured actually predicted protection? We found that if we did 17 different high throughput in vitro assays, we could predict protection in that first mouse model with a 96% accuracy. So this is on the equation, at the bottom of the screen, there's an equation. Protection P is 1 over 1 plus E to the whole bunch of terms. And in those terms, there's a beta and a chi. And the chi is a certain function of the antibody, and beta is the importance of that function. And so that's represented visually above, where a large bar is a predictive and important thing, and a small bar is a less important thing. And so maybe we don't need to do the assays with the tiny bars at the bottom. Blue is a good thing. If the antibody does that, it protects. Red's a bad thing. If the antibody has that, it doesn't protect. So what do we see? The most protective things. Well, first, this is what we used to look at before we started this study. We used to focus on the mechanics of blocking viral entry as measured in the VSV pseudovirus system, and that was sort of middling protective. And then we also looked at where the antibody bound and how tightly it bound at those places. And that looks like it's not as predictive as other things. The most important predictors were the ability to neutralize authentic Ebola virus or filovirus-like particles, and whether it could neutralize completely or if it left some fraction of virus behind, because there was some variability in the epitope. You know, sometimes the epitope has a different conformation and the antibody can only hit it in one. Or the epitope has the presence or absence of a glycan, and one of those means the antibody can or can't bind. So better measures of neutralization were the most important thing. Then everything else combined had to do with what the FC of that antibody did, whether the FC could signal immune clearance of the virus and clearance of the infected cell so that it was no longer a virus factory. And it turned out that those FC-mediated functions were just as predictive in some of the ultimate protective ability of that antibody as neutralization alone. And so what do we figure out? We found better measures of neutralization. Now, the field might have figured those out eventually, but it would have done it one paper at a time. The lab doing that microneutralization assay might have put out a paper saying we think this is better, and then maybe somebody else would have tried it or seen it at a meeting, and then another paper. But because it was run simultaneously side by side with all the other antibodies, and was run on the antibodies from everybody's own lab, and everybody played a role in analyzing that data set, it came into acceptance faster. The other thing is that analysis of the FC was sort of new to us. That had always been a riding variable. We knew IgGs had an FC. We knew the FC did something, but we'd always evaluated our antibody choice based on what the FAB fragment did. And to get that information on the FC, what we needed was one individual person. We needed Galit Alter to say, you haven't looked at this and I have high throughput assays. We needed that one individual voice to propose this to the research collaboration, and we needed it to be run on everyone's antibodies for this new idea to gain acceptance. I think it helped that people saw the importance of these features on their own antibodies from their own lab. And so I think that was the benefit of the broader, larger collaboration, that the individual with the idea was recognized and the utility was demonstrated because everyone had their antibodies in the study, everybody had skin in the game. And so typically what we'd always done is that one lab or a couple labs working together made a panel of antibodies. They would select them by neutralization, in vitro. The ones that would neutralize would go into mice. If they protected mice, it'd go into guinea pigs. Protected guinea pigs get tried in non-human primates. And we found at the end of the day, the end of this analysis, that neutralization alone could find about 60% of the antibodies were ultimately protected. So it works. Hits come out the bottom. But had we evaluated the antibodies based on the effector functions of the FC, we also would have found 60% of the protective antibodies. And sometimes those were the same samples as the neutralizers, and sometimes they were different. And we'd never used FC activity before as a down selector mechanism, but I think there's utility there. And so what we found by building this larger research framework at an international level was that to protect against Ebola virus, we want two things. On the left, we want antibodies that can mechanically block entry. They block the conformational changes that drive the virus in. They block receptor binding. And those are the ones that tend to bind within that black line on the core of the glycoprotein structure. We also want antibodies that can mediate FC clearance of the virus and infected cells. And they tend to bind above the black line on the right in the upper tier. So when I say tier 1 there, I don't mean in the HIV sense. I mean in a wedding cake sense, of the top geographic layer. The ones that bind up there tend to better recruit the FC functions. And so we want both of those features in our ultimate antibody cocktail. And there's one epitope that hits both places, and it's kind of a sweet spot. We call that the head. Antibodies that bind there block receptor binding and tend to recruit the FC functions. And so at the end of the day, the fast track came up with an antibody therapy that was a good proof of concept, and the comprehensive track came up with another one that was had broader activity against more different Ebola viruses. And each of these bring these two features together. They bring neutralizing activity from binding the core and FC-mediating activity by binding the head. And so at the end of that first 5 years of the grant, and it's now been renewed to continue work on Ebola viruses and Lassa viruses and Add and alpha viruses, by putting the body of investigators together with enough funding to do a complementary large study, we were able to write 78 papers and confer 10 PhDs in the research that was found. The group advanced 6 novel therapies and revised the pipeline by which we try to evaluate and mobilize antibodies forward. And that led to antibody therapies for the first time being available to try to treat Ebola virus infection. Now that was August 2019. Spring 2019, I moved my lab to build a cryo-EM and antibody discovery facility, and I'm now at the La Jolla Institute for Immunology, which is shown here. It's a 20 investigator think tank on human disease. We look at infectious disease, we look at cancer, we look at inflammation, and all the different ways in which human immune signaling can be understood and harnessed. This fall, when coronavirus broke out, the Bill and Melinda Gates Foundation and their Therapeutics Accelerator called and asked if I would build a model very similar to what we did for Ebola virus, but now for coronavirus. There would be a need for antibody therapeutics against this novel virus, and there would be a need for a comprehensive effort to compare the molecules being mobilized around the world. And so our workflow is this, that antibodies are being discovered in at least 70 different universities and companies, small startups to multinational corporations around the world. They are all being sent here where their samples are being blinded, and that is to protect intellectual property and protect investment. Everything's given a code name. We aliquot and distribute those antibodies. We do parallel analysis from structural biology, in vitro neutralization, FC activity, which ones are resistant to escape, in vivo modeling. We're going to catalog everything in a publicly available database. And because the antibodies are blinded, this database is going to be built and available real time for researchers around the world to be able to understand the range of activities against spike. Do antibodies against a certain epitope have certain functions? Which ones are most potent and why? Are they most potent because they have the tightest affinity or because they hit a certain footprint? Which sites are complementary and synergistic? From that database of information, we'll be able to down select those cocktails that we want to mobilize for treatment in low and middle income countries. And so an important part of it is that I, the PI, am blinded. I do not know whose antibody is whose. My decisions are based purely on the data and the numbers. But the owners of the antibodies know which are theirs, and they can track them in the study. So they have an opportunity to call the program manager and say, you know, that data point does or doesn't match what our expectation is. We know if we need to revisit that data point. They can also gain information on, is that a molecule that they want to keep investing in? Does it really look hopeful, or is something else looking better? Maybe it's in their own wheelhouse. And everybody retains their own intellectual property, and they can publish and market their antibodies as they wish. We're doing a variety of different assays. In my laboratory, we're doing high resolution structural analysis. We have 2 Titans in X-ray crystallography. We're doing binding analysis at Duke, and this is to different flavors and different emerging mutations in spike. We're looking at the ability of the antibodies to neutralize in both pseudovirus and live virus systems. We're looking at the ability of the antibodies to protect in different in vivo models. We're measuring the FC activities of these different antibodies to find out which of these things correlate with protection, or maybe they don't. The propensity of these antibodies to mediate antibody-dependent enhancement, right? Is there a risk here? Is this something we need to worry about? Do we want an effectorless antibody to minimize that risk? Or do we want to understand those activities so we can improve protection? We're going to study half-life of these antibodies and ABV propensity in vivo using some different animal models. We're going to look and see what emerging mutations in the world cause loss of binding and activity of these antibodies, and which of the antibodies recognize epitopes that are more resistant to escape. This will all be compiled and profiled in this COVID database as blinded information. And it's starting to work. This is what it looks like in my lab this month, with the samples coming in different boxes and the aliquoted and the sampling bound. Important components of the study are the data sharing, that in order to defeat this pandemic, we need immediate access to data to accelerate our efforts, but we must do it in a way in which the intellectual property is also protected. And a collaboration like this will only work if there are benefits to the people contributing the antibody. So there's an opportunity for them to contribute to this global crisis. There is that market of achieving protection in low and middle income countries might be something that they're not in a position to make a large donation of material for. This is a way in which the molecules that they've already discovered, that they are free to mobilize and market as they wish in North America, and Europe, and Asia, can also be used to save lives everywhere around the world. The foundation helped to make that possible. So it's assisting them in doing what they went into science to do in the meantime. We can provide a complete independent data set of lots of different kinds of assays to which they might not have access themselves. And we can gauge the performance of their molecules and standardize assays relative to the others, so they know if it's worth continue investing. We might find a pair, a synergistic pair, that we wouldn't find in other ways. Maybe it exists from 2 different labs. And we also have the opportunity, because it's important for us to reduce the cost, to increase the deliverability of these antibody therapies, to analyze the sequences for problem residues that might complicate manufacturing and to make improvements. And the owner of that antibody, the original donor, gets all that IP too. So if we improve their molecule and make it easier to manufacture and more potent, they own those improvements. And same with FC. And the important thing to note is that the goal of this collaboration is to complement what these groups are trying to do themselves. We are not competing with their discovery effort. We are slower because we are larger, and we are providing them additional data that they can use for their own IND filings and use in their own papers. We are just trying to find a way of helping their molecules move toward the clinic. And if I still have about 5 minutes, I can talk about a new piece of data from the lab. We are keenly interested in whether or not the spike of this virus accumulates a mutation that would knock out one of these individual monoclonal antibody therapeutics. And so when the D614G mutation erupted and then by late March had become dominant on the world, we became quite interested in what it does. So with now 12 million cumulative cases and that curve steepening instead of flattening, that's a lot of human-to-human transmission. And it's perhaps not too surprising that a mutation did erupt, even though the coronavirus does have some proofreading capacity. And this was the work of Betty Korber at Los Alamos National Lab, tracking the presence of the original Wuhan reference strain of the virus in orange, and the emerging point mutation with a G instead of a D at position 614 and spike, and that's blue. It emerged kind of the end of February, early March, and then in the next month had become dominant. Became dominant in Europe and North America. England had what looks like here 2 waves of just the new virus. And if you look at Taiwan, they seem to have cleared the original virus, and then the new blue virus was introduced and caused another outbreak. So a mutation was there. What does it mean? Is it anything we need to worry about? Well, we did pseudovirus neutralization assays in my own lab, and David Montefiore, also in the paper, did lentivirus assays in his lab. And we looked at the ability of viruses carrying the old spike to neutralize in different cell lines, Viro 293T and then also 293T with the addition of the TNPRSS2 protease that the virus uses. And this is how well those viruses infect. But with that single point mutation and spike, they go to higher titer. It is somehow more infectious or more fit, 2 to 3 times in our hands, larger in a lentivirus hand. And so the point of doing this study was because the novel emergent form of the virus actually came with 4 mutations, 1 in spike and 3 somewhere else. And so we wondered if any effect of this could be attributed just to the spike mutation. That's why they did these studies. The 1 point mutation enhances infectivity, so the virus grows 2 to 3 times higher titer. And there are now multiple preprints in bioRxiv that find the very same results. So that's what happens in vitro and in cell lines, what happens in people. And this is also in the paper. This is work studying patients in the Sheffield, United Kingdom, by Thushan de Silva. They look at the CT value, the cycle threshold for diagnostic PCR. The CT value for patients known to be infected with the original D strain are in orange and the new G strain in blue. The average CT value for blue is lower. It needs fewer cycles of amplification to detect a virus in those people. It looks like the virus grows to higher copy number in people as well as in cell culture. So is this something we need to worry about? Well, we looked at San Diegans that had recovered. Now, each of these San Diegans were infected in early to mid-March when both the D and the G viruses were co-circulating. So we don't know which of these strains each of these people was infected with, but we can look at the neutralization of the antibodies and convalescent sera against the orange virus and the blue virus. And so we can look at relative infection of cells on the y-axis and dilutions of antibody on the x-axis from straight up to highly dilute. And we could look at the amount of sera required to neutralize 50% of the pseudoviruses versus 90% of the pseudoviruses. And in every case, there's not a big difference. If anything, in every one of these donors, the blue virus is a little bit to the right. The human convalescent sera is just as good as, or maybe just a smidge better at neutralizing the G than the D. So even though the virus grows to a couple of times higher copy number, you don't need a couple of times more antibody to neutralize it. The human polyclonal response is definitely up to the job. Now that's a limited study. That's the convalescent sera of these 6 people. There are papers now in bioRxiv that have more people and analyzing individual monoclonals. It looks like the monoclonals against the receptor binding domain are not effective. And more studies are needed just to make sure that antibodies or vaccines elicited against the D are still responsive against the G. Now I expect they would be. This is one tiny point mutation across well over 1,000 amino acids. A polyclonal response will probably not be affected until there are many mutations, but an individual monoclonal could, and it's for that reason that we've been trying to keep an eye on this. The other important thing is that the novel virus does not seem to cause a worse patient outcome, at least in this study. This is now 1,000 patients in the Cell paper in Sheffield. And patients infected with G versus D don't have a higher proportion of winding up in the ICU versus being inpatients versus being outpatients. What we don't know is if it triggers an asymptomatic to symptomatic outcome, because if someone was asymptomatic, they didn't go to the hospital to be counted. And it's certainly plausible that if the virus grows to a much higher copy number, there might be a higher incidence of transmission. Now, we haven't actually done the transmission studies. I'm sure those are in progress right now. But what we noticed is that G614 emerged and within a month became globally dominant. And now the global outbreak is with the G virus, and that virus is somehow fitter and stronger. So mutation at this point became globally dominant a month later. And with this much human-to-human transmission that appears to be only increasing instead of decreasing, there is certainly a possibility that there will be another mutation that could emerge. And the need for these vaccines and therapeutics is greater than ever. And so there's very much a need for global collaborations like COVID and like the Global Virus Network to accelerate our ability to find antibodies, vaccines, antiviral drugs, mechanisms of cleaning and eradicating virus. And we must find new ways of working together faster and smarter to discover what we need, de-risk it, and evaluate its activity. And I'd be very happy to answer any questions that you might have.
Thank you so much, Erika. It was really great presentation and exactly consistent with, I would say, the spirit both of these webinars and the Global Virus Network. So thank you very much. That was great. So we can have questions. So maybe you can see the questions. You see, Erika, on the Q&A on your screen. Otherwise, I can you see the
question? Okay. So Moses asks, I wish to ask if a patient is sick with SARS-1, the 2003 virus, and develops antibodies against those. Can those antibodies act as memory cells in case of a recurring infection or mutation? Maybe. There are definite therapeutic antibody candidates that were discovered in the time of SARS-1 and were either found to be cross-reactive or were engineered to have higher potency for the new viruses. So it is conceivable that there is some cross-reactive antibody in people that survive that. And I've been hoping to see some studies of survivors of SARS-CoV-1 and see if they've been exposed to SARS-CoV-2 and how they fared and how their immune response might be different from somebody who was naive and then exposed to SARS-CoV-2. So I don't know the answers to that. And if somebody does, please write in the chat because I think that's a critical question. because we will never be done with coronaviruses. There will probably always be another one. And this one could have seasonal occurrence. It could become as endemic as the common cold coronaviruses. And so it's going to be important to understand how that antibody activity works. Now, it does look like T cells from common cold coronavirus infections can have some cross-reactivity to the novel coronavirus. So presumably T cell responses against SARS-CoV-1, which is even closer to SARS-CoV-2 than OC43 or the other common cold coronaviruses, could have some memory. Shinhee Kim, do you have a good animal model system to study transmissibility of the G14 mutant virus? I think that that virus has, you know, with just the 1 point mutation across a large genome, I think you can use the same model systems. My laboratory specializes in structural and molecular biology, and I guess now an administrative organization of things. We are not animal modelers ourselves, and so I think that is best in the hands of the professionals that are evaluating the different animal models for pathogenicity and transmissibility. And I think all of those studies are being done in parallel in the different BSL-3 labs. Our BSL-3 is under construction and now nearly done, and we will be able to do some modeling, but we are focusing our effort here on the therapeutic antibodies and what we can understand about their half-life and altered affinity for Fc receptors.
And Erika, if I may interrupt just 1 minute, regarding this question of the animal models, and exactly as you said, you really need to have specialized laboratories and obviously in the appropriate infrastructure. Just to mention to the audience that the Global Virus Network has been really making an overall evaluation of the different capacities of different members of the GVN all over the world regarding the animal, the capacity to run animal experiments. So this is something where we can provide for those who are interested some advice. And I forgot to mention this in my introduction, but GVN is very much focused on interaction with industrial partners also. And this is something that we can provide access to these animal facilities. Thank you.
Another question, are you finding differences in G versus D sensitivity for monoclonals? Not yet. Most of the monoclonals being mobilized for therapeutic use are directed against the receptor binding domain, which is a separate location from the G versus D. And the ones that have been analyzed so far and deposited in bioRxiv don't look like they're susceptible. Now, it's certainly plausible there's a different antibody against another site that would be, or if there's some different behavior in a respiratory lung cell line as opposed to a viroRNase 2. I mean, a 293 cell line that a monoclonal could be affected. Dr. Hernandez, are you also testing neutralizing antibodies from recovered patients that may be useful developing new mAbs as well as already developed? Yes, we are. We have intentionally decided to remain out of the therapeutic space in order to remain unconflicted, but we are discovering novel antibodies and we're just going to make them available for research use. There are lots of people looking at new survivors as well, and I think that the field will have an absolute wealth of monoclonal antibodies against this virus, and we'll have a very good understanding because of the number of data points of what epitopes are ideal, which things are synergistic, which do and do not get escaped by different mutations. Dr. Protzer, hello, how are you? The big risk of antibodies not immediately neutralizing is that they of course cause ADE or advanced immunopathology. Any way to avoid this by selecting the antibodies in the cocktail designing vaccine? That is absolutely the critical question. And that is the worry. If we're operating at warp speed and rushing things to clinic and evaluating them, you know, based on in vitro data and novel models that haven't been fully well characterized, that is a real risk that an antibody could, especially for a respiratory disease, increase uptake and accelerate disease. And so I was worried about that. And so there are a couple ways around that. For other viruses, it looks like enhancement occurs when neutralization is suboptimal because the affinity is suboptimal or there isn't a sufficient concentration of antibody to neutralize. And so if one can boost affinity, boost potency, or choose the very best from among 1,000 antibodies, you might have a chance of having neutralization outcompete ADE. You can also mutate the Fc to make them effectorless, to remove affinity for Fc receptors. And so you're operating by only neutralization. Now the plus is you may have removed any risk of ADE. The minus is you've removed protective effects of FC. And I think for this novel virus, we don't yet know which is the right side to err on. And that's why we doubled the size of the consortium to add in all of those experiments, because we just don't know, and we need to know. The very first therapeutics mobilizing forward at this breakneck pace are going to be the most potent neutralizers. But I think that if we need a second generation therapy, if the virus stays around a few years, we really need to understand that FC question. And so across the panel of therapeutic candidates, we are doing broad immune profiling. What are all the different functions that each one of these antibodies elicits? Which are stronger than others? Does that have to do with the epitope on the spike they bind, whether it's the top or the middle of the side? Does the strength of those functions map with the strength of the protection? How important is FC? Do we need FC? And we're also looking at different modifications of the FC. So the therapeutic candidates going forward first, each have a different FC. One has a very standard human IgG1 FC. One has been engineered for a long half-life mutation. Now that makes sense. You want the antibody to be around months instead of weeks, especially if you're gonna use it for prevention. But that long half-life mutation enhances affinity for neonatal FC receptor tenfold. And so that enhances uptake and recycling. But if you're enhancing affinity for the FC, are you also enhancing ADE? And I think we don't have that data yet. And so that's what we're trying to do here on the academic nonprofit level, is just try to build that body of information so that we know it. So we can make sure that if there's a safety issue, we have the information to address that. And we have a novel therapeutic where we can engineer the FC in the right way. Can you please confirm whether COVID-19 could be airborne? I think there's some confusion about the term. It definitely spreads in minuscule respiratory droplets, and they can kind of hang and float in the air. Because it is a membrane enveloped virus, it is not as hardy as something like adenovirus. And I think I've seen studies where something that has a hard protein shell like adenovirus can blow from one village to another. I don't think a membrane enveloped, more fragile virus like SARS-CoV-2 could blow from one village to another, but it could certainly blow from one face to another face. So it's on a smaller scale. The airborne nature is not necessarily my expertise. I think we have people in the GVN that know a lot more about that than I do. Professor Bonagoro, hello, how are you? Is there any data on different binding efficiency for animal ACE2 versus for different SARS-CoV-2 variants? Yes, there is. I don't have that information at my fingertips, but there is a body of information on the range of different coronaviruses similar to this and for what species they have been adapted and evolved. Anonymous presentation, great presentation, thank you. With time being a limiting factor, indeed, do you have a recommendation for the type of antibody constant domain to use based on your previous work? I would look at the work of Galit Alter there. What we did for, and I think it might be different for every virus, and I think it's different for every epitope. So for example, for Ebola virus, we had antibodies that bound the top and antibodies bound the side. And for the ones at the top, they seem to work a little bit more by phagocytosis, and the ones that bind the side tend to work a little bit more by natural killer cells. And that could be different for every kind of viral spike. Coronavirus could be the opposite. But what we did for the VIC, the Ebola VIC, is we had those two groups of outliers. We had the neutralizers that didn't protect, and we had the protectors that didn't neutralize, and it had to do with FC. And so we started engineering the FC to see if we could take the effective ones and break them, and we could take the ones that neutralized and failed to protect and add in FC and see if we can make them better. And indeed you can. But I'm worried that this is going to be particular to each virus and particular to each spike. And so I would not hazard a guess quite yet about what's going to be ideal for SARS-CoV-2. I think nobody knows until we have the data. Dr. Feruse, great talk, thanks. A fraction of patients seem to develop severe illness, not due to viral cytotoxicity, but too much immune response. Indeed, that is true. I heard this happens even after viral titer decreases. Is antibody administration promising to treat those patients? I think antiviral antibody will be most effective early in infection, where its job is to knock down the viral load. Latent disease course, the severe acute respiratory distress, is an overactive inflammatory response in the lung. And that is caused by the immune response that is not caused by the virus. So I think the utility of an antiviral antibody at that point in time is lower. But there is an opportunity for a different kind of antibody therapy. So antibodies, that, for example, against IL-6 that upregulate an immune response can tend to damp that down. And those antibodies discovered 15 years ago in studies of fundamental innate immunobiology are now being evaluated in clinical trials in 4 continents. And so there's, I think, early in infection or to prevent infection, you want an antibody against the viral spike. Later in disease course, to control the inflammatory disease, you probably want a steroid or you want an antibody therapy against those inflammation modulators. You mentioned polyclonal treatment. Do you have an idea of the number of different antibodies that would be optimal and how this affects clinical development and safety? So polyclonal treatment, there's, I don't know, 1 million different antibodies in it. And the benefit of that is that they hit so many different epitopes. And so it's hard to escape. And it's a good thing to mobilize very early in infection. We haven't found your monoclonals yet. But we don't know always in an individual polyclonal sera what is the most important factor and which antibodies are just along for the ride or duds. So I don't have an idea about which particular antibodies in the polyclonal are effective, but it does look like, as a proof of concept, polyclonal has achieved benefit in some patients. Thank you for an amazing presentation. Oh, thank you very much. Antibody potency. What of their tested protective durability? Do you mean durability in terms of whether or not they can survive seasonal drift and mutation, or a mutation and escape within a patient? I don't know. We might not have that data until the clinical studies are actually done. You can imagine a clinical study of, say, healthcare workers that had a high likelihood of being exposed. And if you've given them prophylactic antibody in advance of vaccine availability, and you look and see who, if some of them got infected anyway, and what the strain of virus was that infected them, you could know if there was something that had escaped that antibody or if it was something else. If it had to do with antibody half-life and pharmacokinetics. There's a lot of PK studies being done right now in animal models to understand tissue distribution of the antibody therapies and how long they last.
Okay, well, thank you so much. That was great, really, really great. Thank you so much, Erika. Thank you to all of you for attending these webinars. And you have the possibility to be in contact with the Global Virus Network very easily. We have the website. You can contact us whenever you want. We can really provide this connection between scientists, and really Erika has so well illustrated the importance of these international collaborations. And actually this is really a new mode of science, really much more fitted with fighting against this pandemic. So thank you so much, and we wish you, all of you, a very good day. Thank you, Erika. Bye-bye.